Peter, I think you can kick us off.
Okay, thank you very much. Good morning, good afternoon, and good evening, wherever you are. I presume that many different people and time zones present here. I'm Peter van Daal. I'm Head of Nutrition Science and Advocacy at DSM. On behalf of my colleagues at DSM and myself, a warm welcome to all of you. Happy to see that we have a lot of interest in this afternoon's presentation by Professor Hania Szajewska on human milk oligosaccharides. I'm pretty sure we will learn a lot on this interesting, functional and nutritionally active compound class of compounds. Providing the best start in life and making sure that infant nutrition is optimal is really a mission we are proud at DSM to support. Now, as mentioned, we're very pleased to be today joined by Professor Hania Szajewska. I have known Hania for quite some years, and I can but imagine that Hania will be a very, very passionate speaker. She has a lot of insight. She's a dedicated researcher and a dedicated pediatrician since many, many years, and infant nutrition and sharing her knowledge about it is a passion I truly enjoy every time she presents. Now, before we're going to give the floor to Hania, I would like to share a few housekeeping rules. If you have questions, please put them in the chat box. We hope we will be able to answer them all, but if we will not be able to do that, you will get definitely an answer after this presentation. And we will also, for those who want to see part of the presentation, make sure that you get a summary of some slides plus a kind of a summary of the key points sent to all who attend this session. You can open and close the cockpit using the red arrow. So if at some point in time you need to leave, you can do that by pushing on this red arrow. But without further ado, I want to give the floor to Hania. And I'm sure, Hania, you will enlighten us on the HMOs in the next 30-45 minutes. Without further ado, Hania, over to you. Thank you.
Thank you a lot. Thank you very much, Peter. I hope you can hear me and I hope you can see my presentation. So good whatever time of the day is for you. It's really my pleasure to be here with you and welcome to today's webinar on human milk oligosaccharides, current knowledge and aspects to be clarified. Peter was so kind as to introduce myself. I would just add that my scientific heart belongs to ESPGHAN, European Society for Pediatric Gastroenterology, Hepatology and Nutrition, and more recently also to International Scientific Association for Probiotics and for Prebiotics. And I have to admit, I really like evidence-based medicine. Let me start my presentation with one of my favorite books, which is Animal Farm by George Orwell. If you have read this book, you probably remember this famous quote, "All animals are equal, but some animals are more equal than others." And let me paraphrase it and say, all milks are equal, but some milks are more equal than others. I'm a pediatrician, and as pediatricians, we do promote breastfeeding. We do promote breast milk, and there are many reasons for that. And here is a quote from BMJ, British Medical Journal, which I like very much. "Formula milk is just a food, whereas breast milk is a complex living nutritional fluid that contains antibodies, enzymes, and hormones, all of which have health benefit." As a matter of fact, if you look at this slide and you will compare formula on the right side and breast milk on the left side, you will see there are much more differences than what has been said, shown on previous slide. A lot of those are live cells, a lot of those are components with some immune properties. All of those contributes to the benefits of breastfeeding, and one of the strongest evidence that we have when it comes to benefits of breastfeeding is protection against childhood infections. And many components contribute to that, but among those components are also human milk oligosaccharides, which I will discuss today in more detail. This is the third largest solid component after lactose and after lipids. There is more of human milk oligosaccharides than there are proteins. Human milk oligosaccharides, HMO, are known for years, but they are discovered again. They are known for years because they were first described in the 19th century, and it's already in the middle of the 20th century that it was possible to isolate and characterize about 100 different HMOs. But now there is really like a huge interest in HMO, and you may ask yourself, like I'm asking myself, as people are asking, what is behind this current interest in HMO? Now it's of course lockdown, but before that, at almost every meeting, pediatric meeting at least, there was a lot of sessions about HMOs, and also there's a lot of webinars. So this is a valid question. What is behind this current interest in HMO? I think there are a few reasons, and allow me just to elaborate on them. First of all, advances in analysis of human milk oligosaccharides. It's much easier to study this human milk oligosaccharide nowadays than it was previously. The second reason is progress in biotechnology, which nowadays allows to produce at least some HMOs in such quantities that they can be used and they are being added also to infant formulas. And last but not least, there are formal approvals for organizations such as EFSA, European Food Safety Authority, and FDA, Food and Drug Administration, which confirm safety of, here it's only written 2FL and LRNT, but there are more coming, when added alone or in combination to infant, follow-on, and young child formula. So this explains, I think these three factors explain why there is such an interest in HMO. So what I would like to discuss today, I would like to tell a little bit about basic facts about HMO. I will talk about HMO present in breast milk and health-related effects, and I will talk about findings mainly from human observational studies. Then I will discuss specific HMOs which are manufactured, which are added to infant formulas, and I will talk about findings from randomized controlled trials, and then I will discuss some aspects to be clarified. I'm a pediatrician, I'm a clinician, so very much it will be clinically oriented, but perhaps during the Q&A session, we can also address some other types of studies. So let me first start with some basic facts. What are HMOs? So HMOs are complex carbohydrates composed of the 5 monosaccharides, such as glucose, galactose, N-acetylglucosamine, fucose, and sialic acid. On the right side, you can see there's different structures because all these carbohydrates are linked in various ways. But all HMOs carry lactose. For one reason or another, my pointer doesn't work, but doesn't make any difference. For each one of them, you will see that each contains lactose. And then when it comes to biosynthesis, there are 4 processes which plays a role. So this lactose can be elongated, the structure can be drenched. You may see the fucosylation, which means that to this basic structure, which is a lactose, fucose is added in alpha-1,2 linkage, and then you have 2 fucose lactose, or you may have the 4th process, which calls sialylation, which means that to these basic structures, to lactose, sialic acid is added. Here on this slide, I summarize some major HMOs in human milk. There are fucosylated HMOs, and the examples include 2-fucosylactose, 2FL, or 3FL, 3-fucosylactose. There are sialylated HMOs. An example is 3SL or 6SL. And finally, there are also non-fucosylated neutral HMOs. And one example, and you will hear about it also during my presentation, is LNNT, which stands for lacto N-neotetrose. Altogether, at the moment, there are at least 150, probably even 200 structurally different HMOs. And I already mentioned that for the biosynthesis, all this branching is important. And there are very specific branching and very specific linkages which are important to the function, which means that not all HMOs are the same. And allow me to compare to Lego bricks. So each of these bricks corresponds to one of the monosaccharides. And then you can combine these Lego bricks in various ways, depending on the imagination, maybe not yours, but your children. But anyway, it may result, depending how you link them, how you add one to another, it may end up a tower, car, or a house, and the function of each one of them will be different. And it's exactly the same when we are speaking about human milk oligosaccharide. Depending on these linkages, the function may be different. And it's very important that the function depends on the structure. One of the questions which is often asked is the composition of HMOs, all mothers, the same? And the answer is no. Each mother produces a specific HMO set, and which is as unique as her own fingerprints. So then the next question could be, what are the factors affecting HMO composition? And again, you will see on this slide, on the next slide, that there's a number of factors which are affecting HMO composition, and as so often, genetic factors play a role. HMO pattern depends very much on the expression of two genes, FUT2, which stands for fucosyl-transferase, this is so-called secretor gene, and FUT3, which is a so-called Lewis gene. And then, depending on the activities of each one of those genes, there are 4 different possibilities. You may have active FUT2 gene and FUT3, and then you will have Lewis positive secretors mothers, and you will have on the right side, on the bottom, the other extreme, that both of these genes will not be active, and you will have Lewis negative non-secretors mothers, and 2 other options in between. So genetics very much affects HMO composition. But this is not the only, and what is important that about 80% of the European and American women are secretors. There are studies showing that there are geographical differences, but this is what is typical for European and American. Most of them are secretors. What does it mean? That they secrete all HMOs. Okay, what are the other factors which contribute to which affect the HMO composition? On this slide, you can see that that depends also on the lactation status, early versus later lactation. It depends on the delivery, term versus preterm delivery. The data are conflicting, and one of the reasons that the data may be conflicting is because of the methods which are applied are not standardized. So there are differences between various laboratories. Maternal diet, people are really very much interested in whether or not maternal diet, it's not very well studied still, but a number of teams is working on that. There are also other factors which are considered, like for example, gender, microbiota, with a huge baseline microbiota, which is a huge question mark because at the moment we don't know. And as I said previously, commenting on term versus preterm delivery, methods applied, there is no standardization. So this may explain the reason why what we read in the literature is not sometimes conflicting data. So further studies are needed, and some people are working on it. Now let me move to HMO metabolism. One important feature of HMO is that they are only minimally digested. Almost all HMO reach the distal small intestine and colon intact. It's only approximately 1% of the HMO which is absorbed and excreted with urine. So if they are only minimally digested, and there's so much of this HMO, more than proteins, then the question is, what is the function of what are the effects of HMO? And on this slide, I summarize those 4 major effects which are often discussed in the literature. And let me discuss each one of these effects in more detail. Starting with an effect of HMOs being prebiotics. What is a prebiotic? Prebiotic is a substrate that is selectively utilized by host microorganisms, conferring a health benefit. Prebiotic effects are structure specific, and different HMOs have different effects on microbial community. But what is important, that HMOs promote the growth of beneficial bacteria, bifidobacteria. If there is one major reason, there are many, of course, between breastfed and formula-fed infants, but if there are one major reason, it relates to microbiota. A microbiota of infants, gut microbiota of infants who are fed human milk, they are very rich in bifidobacteria. And this is because human milk delivers human milk oligosaccharides, which are then used by bacteria, mainly bifidobacteria. And then you have this Bifidobacterium longum, subspecies infantis, Bifidobacterium bifidum, Bifidobacterium breve. Bifidobacteria subspecies use HMOs differently, but they can use this unlike humans being. And it's a tremendous effect, beneficial effect of this Bifidobacteria, which is really important and contributes, I will repeat it again, to the differences between breastfed and formula-fed infants when it comes to gut microbiota. So this was the HMO effect as prebiotics. Another important effect is they have anti-adhesive, antimicrobial effects. As my pointer doesn't work, then on the left side of this slide, you can see pathogens which are attaching to receptors on the surface of the epithelial cells. This is needed for initiation of any inflammatory reaction. And these receptors, which you can see here on the surface of the epithelial cells, are very similar to, or HMOs are very similar to those receptors. So if you have any pathogens in your gastrointestinal tract, they may attach to HMOs, and they may serve like a soluble glycan receptor decoys. And if they attach to each other, they will not be able to attach to these receptors on the epithelial cell, and they will be excreted with stools, protecting in this way infants against gastrointestinal or any kind of infection. So these anti-adhesive, antimicrobial effects are very important. Another postulated HMO effect is that they serve as intestinal epithelial cell modulators. So HMO directly affect intestinal epithelial cells and modulate their gene expression, which can lead, and you can see on this figure here, leads to the changes in cell surface glycans and other cell responses. In other words, the structure of this receptor will be changed to such an extent that the pathogen will not be able to attach to this receptor, again contributing to these protective effects of human milk. And finally, another postulated effect is that they may serve as immune cell modulators. So HMO can modulate lymphocyte cytokine production, potentially leading to a more balanced Th1, Th2 response. And this is important because if you have the dominance of Th2 response, it increases, contributes to the allergy risk. If you balance it, you can have, this may have an effect on the reduction of the risk of allergy. So my summary number 1 for this part of my presentation, where I did try to summarize some basic facts on HMOs, is HMOs are the third most abundant solid component of human milk. HMOs are not digested. HMOs are structure determines HMO function, so not all HMOs do the same. And I also briefly discussed postulated effects, which include the prebiotic effect, that they serve as anti-adhesive, antimicrobials, intestinal epithelial cell modulators, and last but not least, immune modulators. This brings me to the second part of my presentation, and now I would like to discuss HMOs which are present in breast milk and some health-related effects. I am a clinician, so I will mainly focus on findings from observational studies. And as you can see here, there's a number of conditions, health outcomes, for which this links association between specific HMOs and health outcomes were studied. Let me give you just some of the examples. I will start with infections, particularly diarrheal diseases. Diarrhea is really still a problem, especially in low and middle income countries. Deaths in children below 5 years of age, 500,000 per year, much less than it used to be, but still in some parts of the world, this is a huge problem. And there are studies, and I'm just showing one example, that there is a link between levels of 13 HMOs, here 2FL, 2-fucosyl-lactose, and the risk of diarrhea, Campylobacter diarrhea. So on this slide, you can see that if you have a high levels of 2FL, then you have the much lower risk of Campylobacter diarrhea. The same has been shown in a similar population, has been shown also for enteropathogenic E. coli. There are also some data, but more like laboratory studies showing that also when it comes to viral infections like rotavirus and norovirus, that it can also protect. Another condition which I would like to discuss is neonatal sepsis. It's a major global health problem and contributes significantly to infant morbidity and mortality. You have to be aware that more children are dying of sepsis than they are dying of all cancers together. So this is really a tremendous problem. And then the question is there any role for HMOs or specific HMO and the neonatal sepsis? This is a very recent study published in the American Journal of Clinical Nutrition this year, which shows that higher concentration of one of these fucosylated HMO contributed to better survival. So another example that there are links, association between HMO in human milk and some health outcomes. Another condition which I would like to discuss is allergy. In high-income countries, 1 in 10 people live with a food allergy, with the highest prevalence amongst infants and young children. And of course, people are trying to understand what is behind it and what can be done to reduce this allergy. So I'm just showing you some of these observational cohort studies, human studies, which showed associations, links between levels of 13 human milk oligosaccharides and, like, for example, the Finnish study showing that there was a link between high level of one of these fucosylated HMOs and decreased likelihood of cow's milk allergy. Another study which shows that high levels of 2FL, 2-fucosyl-lactose, potentially reduces the risk of IgE allergy in C-section born infants at high risk for allergy. Allow me to comment on this C-section. This is important because the number of C-sections is increasing in many countries, and we know that C-section increases the risk of allergy, also the risk of obesity. Another study showing that the specific profile of HMO is linked to the lower rate of food sensitization. Food sensitization is not the same as allergy, and not every food sensitization will end up in allergy. However, many children with food sensitization later on develop allergy. So this is an important finding. And last but not least, very recent study published in JACI, Journal of Allergy and Clinical Immunology, which shows that there are specific profiles of HMO which may contribute to increased allergy or to decreased allergy. Of course, more studies are needed. These are observational studies showing association links, but definitely there are some links between some specific HMOs and this outcome. Another condition which I would like to discuss briefly, necrotizing enterocolitis. You don't have to be a pediatrician. You don't have to be a neonatologist to know that this infant, this is a preterm infant, has a very, very severe condition. And this is necrotizing enterocolitis. 30% of those children will die. And even if they survive, approximately half of those who will survive will have neurodevelopment consequences. So of course, we want to reduce this risk of necrotizing enterocolitis. And here, the #1 prevention measure is of course human milk oligosaccharides. but people want to understand what is in this human milk which makes such a big difference, and whether, because also some breastfed infants, they still develop necrotizing enterocolitis, so there might be some difference between different mothers. And here are the studies. One exception that I'm also showing you, the animal model, but it started with the animal model, but then later on it was confirmed in human studies that the concentration of one of these human milk oligosaccharides, in particular DSLNT, diacylylolacto and tetraose, that the concentrations were consistently lower in necrotizing enterocolitis cases. So again, there are links between certain HMOs and health outcomes. Another one, growth and body composition. A number of studies showing that some of the HMOs, like fucosylated 2FL, but also 3SL and 6SL, they may have an impact on growth. And what is in one of those studies, what has been also shown that the ratio may be important. And another condition, neurodevelopment and cognition. This year, the study was published that early exposure to 2FL is important for enhancing infant learning and memory. And here, this study showed that the timing is important, that the early exposure was important. So for this part of my presentation, I did try to show you that there are specific HMO in breast milk which are linked to certain health outcomes. And I discuss it in the context of infection, sepsis, allergy, necrotizing enterocolitis, growth, body composition, and neurodevelopment and cognition. Why it's so important, this kind of studies? It's important because establishing a link may lead to intervention strategies. Once we know which factors are responsible, we may think about next steps, like isolating them or manufacturing those particular HMOs and then using them for preventive or therapeutic strategies, for prevention of or for therapy. And this brings me to the third part of my presentation, that I would like to discuss specific HMOs added to infant formulas. And I told you that I really like evidence-based medicine. That's why I will focus on findings from randomized controlled trials, because they're always, there for any prevention of therapeutic studies, this is the highest level of evidence, randomized controlled trials. I already showed you this slide, what is behind the current interest in HMO. And I told you the progress in biotechnology, which nowadays allows to produce at least some HMOs to be added to infant formula. And I think more or less it started in 2015 that companies started to add once they became available, and probably most of the studies are on with those two HMOs, 2FL and LLNT, so 2-fucosylactose and lacto-N tetraose. These are some of these randomized controlled trials which were already published. Again, I'm focusing on randomized controlled trials because the strongest evidence always comes from randomized controlled trials. And let me summarize what are the data when we are discussing what is published. So first of all, there are infant formula with 2FL with or without LNT. And the studies show that they are safe and well tolerated and support normal growth. Let me comment on it. These kind of studies are extremely important because if people want to sell any kind of formula, regulators also require it, but also the pediatric community or academia will require that it has to be documented that the growth of those children fed infant formula supplemented with anything new should be similar to those children who are unsupplemented, and more important, similar to that of breastfed infants. So this is extremely important, and without those such studies, EFSA or FDA would never provide this statement regarding the safety of these formulas. There are also studies showing that 2FL supplemented infant formula exhibit immune biomarkers similar to that of breastfed infant. This is again very important because not only that we want to know that the growth is normal, but also that the metabolic responses, immune markers are similar to that of breastfed infants as possible. There are studies showing that 2FL and LNNT, when added to infant formula, shifts the gut microbiota closer to that of breastfed infants. And I already told you how important it is, especially the presence of bifidobacteria. And one of the major differences that we see between the breastfed and formula fed infants when it comes to microbiota is with regard to bifidobacteria. And last but not least is that the addition of 2FL and LNNT is associated with reduced morbidity and antibiotic use. And let me comment a little bit on these outcomes. This one of the studies which show that infant formula with 2FL and LNNT added results in lower incidence of bronchitis. It results in lower respiratory infection when it was evaluated from 0 to 12 months. It results in reduced use of antibiotics and reduced use of antipyretics, at least during the first 4 months of life. Let me just comment on this outcome. Reduced use of antibiotics. What has been shown that this formula with 2FL and LNNT reduces the use of antibiotics by 31%. 31% reduction. Why I think it's so important, and I really always focus on it. And this is that, of course, we all agree that antibiotics save life, and every now and then they are really needed. But there's a problem with overprescription of antibiotics. These are recent data from the United States. 25% of hospitalized children get antibiotics they don't need. Recent data, 2020, from New Zealand, 82% of children receive at least one course of antibiotics during the first 2 years of life. So any intervention that would reduce the use of antibiotics is worth studies, is worth considering. Why it's so important? Because we know that early life antibiotic exposure is associated with increased risk of some of the diseases. What do I mean by early life? It's during pregnancy and during the first 2 years of life. And together with my colleagues from my department, we looked at these effects and we showed that there is association with this early antibiotic exposure and increased risk of allergic diseases, increased risk of obesity. There are other studies showing that there is increased risk of celiac disease. Data on autism spectrum disorders are not so clear. But at least for those two outcomes, allergy and obesity, we know that early life antibiotics may be really harmful. That's why I showed you that I'm focusing so much on this particular outcome. So I showed you the data of 2FL and LNNT, infant formula supplemented with those two, but thereafter those two HMOs added to infant formula, a number of new HMOs products are now in the commercial pipeline, and there are many of those, probably there will be some new. And there are also further trials which are underway. Like, for example, if you go to clinicaltrials.gov, which is a registry of clinical trials, you can see that there are randomized control trials, large randomized control trials with few hundred participants with 5 HMOs. Of course, you need, first of all, you have to recruit these 500 children, and then you have to follow them usually for at least 1 year. So it takes time, but we all of us are looking forward to see the results. So my summary number 3 for this part of my presentation is with regard to infant formula supplemented with select HMO, that these are safe, well tolerated, and functional benefits are likely. And I showed you what are the potential benefits, and some of them have been already documented. And this brings me to the final part of my presentation, aspects to be clarified. As George Bernard Shaw said, science never solves the problem without creating 10 more. By the way, this is what makes science so interesting. But anyway, there are some aspects to be clarified. And I just summarize some of those aspects which I think that need to be clarified. There are probably many more, but let me just comment a little bit on those. Have we exhausted the choice of HMO, or are there other HMOs to be added? We started with with 2FL, LLNT, there are now few more, and you know there are 150, 200 which are in human milk. Of course, we will never have all of them, but it's really important to see what would be the mixture, what would be really the most effective. Which mixture should be the most effective? And this is my second question. How much HMO should be added and how long it should be given? At the moment, it's usually what is done, and it applies to any kind of clinical trials, that if you want to have the same effects as has been seen in clinical trials, you should just follow exactly the same dosage and exactly the same duration of intervention. But of course, future studies might probably clarify it even more. How to identify the populations which will benefit most of adding HMO? And again, let me comment on it. This is really important because, for example, if you do study in Europe, when infants generally, I'm not speaking, of course, there are some countries where the situation is better or worse, but generally, European population is healthy. So in clinical trials, it's every now and then, it's difficult to show in this relatively healthy population that you can even make it more healthier. But in every population, even in high-income countries, you may identify the populations which are likely to benefit or more likely to benefit. I will just give you an example. For example, children born by C-section, and I told you all this association with C-section and allergy, C-section and obesity. So that might be that those populations will benefit much more of adding HMO. What are the long-term effects of adding HMO? Of course, this is a new kid on the block. So we need this long-term data, of course, and people are working on it. And what should be indication, which of course it's very linked to the identification of the population which will benefit, but also it's linked to the new indication. When I was working on this presentation, I looked at the clinical trials go, what are the registered trials, and you can see people working not only on infant formulas with added HMOs, but also with HMOs which will be given to older children or even to adults. And the conditions being studied include IBS, irritable bowel syndrome, IBD, inflammatory bowel disease, allergy, or chronic diarrhea. There is a huge question mark at the moment because we still have to wait for the results. But there is interest not only in HMOs added to infant formula, but also in other populations and in other indications. So this brings me to the end of my presentation. I know that the audience is likely to remember only 3 things from each presentation or speech. So I did try to summarize what I think are the 3 things to remember. So first, HMOs are a group of complex carbohydrates that are highly abundant in human milk. But not all HMOs do the same. So the structure is really important for the function. And message number 2 is specific HMOs found in breast milk may contribute to health outcomes in infancy and later on. And we discussed that establishing NLE is important because they may lead to intervention strategies. And my final take-home message is specific HMOs, when added to infant formulas, are safe and well tolerated. Further studies are needed, but functional benefits are likely. And I showed you already some of those functional benefits. And my final quote is, make sure you have finished speaking before your audience has finished listening. I did my best. Thank you very much for your attention.
Thank you so much, Hania, for an excellent introduction here to a lot of what's going on in HMOs. My name is Todd Hansen. We have a number of questions. We'll try and moderate that between Hania and also Louise Viksnes, who is our head of HMO biology at DSM. The first question that has come here goes back to the structure of the HMO and comments on the lactose component. Louise, can you comment on lactose intolerance and HMOs given the structure composition?
Yes, for sure. So even though you have, as also Hania beautifully explained, so also thank you, Hania, it was really great presentation. So even though you have lactose, you have structures added to the lactose, and as was also explained, so we as humans cannot use the structures, which means that it reaches the intestine where specific bacteria utilizes it. And this was also shown by some of the bacteria, for example, Bifidobacterium infantis, that takes in the structure, hence there is no lactose released. We have actually, in a clinical trial in IBS patients, looked at this also for checking lactose intolerance and if it was okay tolerated for these IBS patients, and we did not see any problem related to this.
A second question here, Hania, is actually more of a consumer or a user perception of HMOs. Any reflections based on your presence in the hospital and with mothers on how are they thinking about HMOs, and are they aware of it at this point when you see them in the hospital?
Well, as I already mentioned, HMOs are known for years, but discovered again. I think HMOs were not so very, I mean, it's not something, the concept was not very familiar to parents, probably more to physicians, but it's changing rapidly. And I did try to explain why it's changing rapidly, because now it's being added to infant formula. So everyone goes back and starts to learn about HMOs present in human milk. And this infant formula supplemented with some of these HMOs, mainly with 2FL and LLNT, are relatively recent, recently have been available on the market, at least in my country. So it's like, you know, that you need time to get used to it. I think that the concept at the moment, the concept of probiotics and prebiotics is much better known, but it's changing very rapidly. And of course, the fact that it's present in human milk makes it interesting for parents. Of course, we do promote breastfeeding as much as possible. And I have to say, we are not as successful as Scandinavian countries, but we are really working very hard on it for good reasons.
I could give you know, a marketer's view on this as well. We did about a year ago a piece of consumer research with parents that had kids 3 and younger. The reason for that was to try and capture the first years of HMOs in infant formula since the first launches in the US and Spain and Hong Kong. And we actually saw at that point that about 47% of parents with kids 3 and younger already had noticed the HMO term and started to research what that was for. So it's an interesting topic that the parents are also picking up.
Yeah, and probably the one way to learn about it goes to these parents, you know, or this social media and things like that to try to learn. I'm not the one who is using social media, so it's more like a discussion in front of the parents. But I think that one has to be aware that what is important for the parents is what will be the benefit for my children. So that's why I was focusing so much on the reduced use of antibiotics, because I really think for each addition to infant formula, everything that will reduce the use of antibiotics is worth considering. And I really do hope that future studies will look at this outcome. It's extremely important.
Great. There's another question here from Matt around how does HMO composition change over time or stage of lactation? Louisa or Hania, you could both talk to this, I know. Do you want to go, Louisa? No?
Yes, I just needed to unmute. So yes, for sure. I think first of all, we also need to address what Hania said during the presentation to fully understand changes in structures that the method, the way that we measure the HMOs, because you could think of, let's take 2FL, which is the most abundant in secreted breast milk. So if you measure that at one institute and then measure this at another institute, it could be different concentrations. But taking this into account, we have looked at all the literatures that have been published within quantification of HMOs. And here, if you look at the range, so have a range at certain times, what we can see for sure that what you see in the colostrum is different what you see in the transit milk and also in the mature milk. So for sure, the structures changes over time. But of course, we may never learn, but we could get close to what is kind of the reality. As Hania also said, that the HMOs in mothers is a little bit like the fingerprint. Maybe you can also add something, Hania.
I think that you have covered it fully.
There's a question for you as well, specifically, Hania, and I know, the question here is, a lot of research is on the most abundant HMOs. Do you have any insights as to whether people are looking at less abundant HMOs?
Well, there are 150 different HMOs, and not all of them are being studied. This is my understanding from of the literature. And probably they will never, all of them will be studied, but definitely the interest is not only about those 2 or 3 which we are mainly discussing at the moment, but not all of them are being studied exactly the same way, that's for sure. Even if you look at the studies, it's usually the set of various 18 or probably 20 various HMOs which are being studied. Perhaps Luisa can comment on it, but this is my reading of the literature.
I think that's correct. And then occasionally there is probably one of the small ones popping up. For example, DSLNT that you talked about is far outside.
Yeah. Well, but this DSLNT, I think it was a really like a link of various or set of various experiments, starting with in vitro studies and animal studies, and then observational studies. And my understanding is that we are waiting for randomized control trial. So this was a way of thinking, starting with that not all infants were breastfed, that some of them, even if they are breastfed, they do receive human milk, not breastfed, but they do receive preterm infants, they do receive human breast milk, but they still develop necrotizing enterocolitis. So the interest was why it is so that some children still develop. What is the difference between? So I think it was really nice way of thinking about how to do studies. And of course, we know now that it's not the only one, that some others, human milk oligosaccharides, also may play a role. So I think we are just at the beginning of a fascinating, I would say, fascinating journey. But really, we are just starting, which is nice.
We have a follow-up question, I think that somewhat related to this one, which is there was a chart in here on 2FL and its relation to cognitive development. Are there other HMOs that are expected to be in that range? Is a question here. Luisa, do you want to comment on that?
Yes, so for sure there has been studies, both observational and also preclinical study looking at 2FL, but also studies have looked at some of the sialated structures, as Hania also mentioned these structures, so both 3SL and 6SL. There has been an observational study where they have shown, unfortunately, these infants died really early, so they could actually look at brain tissue, and they saw, compared to formula fat, that there were more sialic acid in the brain tissue compared to the formula, which could imply or suggest that these structures, some of these structures, end up in the tissue in the brain. Also, preclinical studies have looked at these structures, also looking at cognition, memory, etc. But maybe also you have more to add, Hania?
No, I think you're absolutely correct by saying, and there are animal studies also. I think there at the moment, when it comes to cognition, it's probably more from animal studies than from the human studies. So we will see. And I think it's also important to underline that when we are talking about these human studies, these are observational studies. We are talking about HMOs present in human milk and what are the effects. We are not talking, at least at the moment, about HMOs being added to infant formulas, and but this would be also extremely interesting to see. And again, can we expect huge changes? Well, that's another issue. They might be statistically significant difference, but what is important for us is whether it's clinically important. That's why I go back again to this use of antibiotic, because reduction by 31% really makes a difference. It really makes a difference for parents, you know, from the public perspective, for health insurance. So I think it's really something worth studying. I would say even if it's the only effect, that would be already worth promoting.
And with those words, I suggest we wrap it up. Thank you so much to our audience who has taken out an hour of valuable time to listen to a topic near and dear to our hearts here, the HMOs. And thank you so much, Hania, for giving this excellent presentation. Thank you so much. I am sure the audience here has appreciated it. And to everyone out there, should there be questions that you felt you didn't get answered today, or if you want to follow up in deeper discussions about HMOs, the DSM team is definitely here to discuss going forward, and the addresses are here. A short version, summary version of Hania's presentation will be made available in the next coming days for you who have participated here. Thank you so much, and have a good afternoon or evening, depending on where you are. Thank you.